Hi there. I'm going to be talking about how competition may shape variation in metabolic rates. And this is some work that I did during my PhD with co-authors Dustin Marshall, Matthew Hall, and Craig White at Monash University. And so metabolic rates reflect the pace at which organisms use, transform, and expend energy to sustain life. And metabolic rates co-vary with many key components of fitness, such as survival, growth, longevity, and reproduction. And so it is likely to be a target of selection. And so metabolic rates also reflect the pace of life. And so previous studies have shown that lower metabolic rates are associated with slower growth rates, later onset of reproduction, and longer lifespans. Whereas higher metabolic rates are associated with faster growth, earlier onset of reproduction, and shorter lifespans. And so metabolic rates are somewhat predictable. So for example, metabolic theory shows these classic allometric scaling relationships, which are widespread. So for example, the slope of the relationship between mass and metabolic rate on a log-log scale is generally less than 1. And over the last century, metabolic theory has explored this origin and maintenance of metabolic scaling relationships all the way from single-cell organisms up to community levels. However, this seemingly strong scaling relationship also conceals a great deal of variation that currently cannot be explained by metabolic theory. So for example, we know that species of the same size can differ by several-fold in their metabolic rates. But then even if we look within a single species, basal metabolic rates can vary across seasons, among populations, but then also even within a single individual, such as before and after reproduction in mice. And so this within species variation in metabolism persists, and basal and standard metabolic rates may even vary by up to 3-fold within a single species after a holding mass and temperature constant. And so we want to understand what drives this variation. We know that metabolic theory currently account for this variation, so we look to microevolutionary processes which may help to explain why metabolic rate varies within species. And so we do have some nice examples of studies that have measured selection on metabolic rate under natural conditions. So we have a study which found negative directional selection on metabolic rate in garden snails, positive correlational selection on body mass and metabolic rates in a common lizard. And then our work recently showed that we found negative correlational selection on metabolic rates for a marine bryozoan. However, a classic tenet of evolutionary theory is that selection should deplete genetic and therefore phenotypic variation, so that heritable traits will become less genetically variable over time. And so we know that metabolic rates can be both heritable and subject to selection, but they vary enormously within populations and even among individuals of the same species. And so the question still remains, how can we use evolutionary theory to explain the maintenance of variation in metabolic rates? And so we know that selection can vary across environments, even across very small spatial and temporal scales. So we wanted to explore this further with regards to selection on metabolic rates and how it might be tied to the pace of life. And so that formed the motivation of this study. So we wanted to understand how does selection on metabolic rate vary across competition environments. And so we did this using the marine invertebrate, Bugula neratina, which is a common subtidal bryozoan that experiences both intense among and within species competition for things such as space, oxygen, and resources. And so we anticipate that both intra and interspecific competition will exert strong context-dependent selection on metabolic rates. And so with regards to the traits of offspring size and metabolic rate, we measured the opportunity for selection. So the opportunity for selection is essentially the amount of variation in relative fitness and it gives us an idea of the maximum potential opportunity for selection that could occur in any given environment. We then wanted to look at the direction and strength of selection acting on a trait or combination of traits. And then lastly, we measured the intensity of selection. So we wanted to know the overall strength of selection acting on all combinations of traits in each environment. Okay, so our study system, Bugle and Erytina, has a complex life history. So the adult colony releases free-swimming larvae into the plankton. The larvae soon settle and undergo metamorphosis to this feeding juvenile stage. And so I measured the mass of individual larvae, and then their metabolic rate, which I measured as the rate of oxygen consumption, or VO2, at 2 hours post-settlement and 24 hours post-settlement. And so I'll refer to these two stages as MRE, which is metabolic rate early, and metabolic rate late, or MRL. Okay, so I took individuals of known mass and metabolic rate, and then I randomly assigned them to one of 3 competition treatments. So firstly, we had a no competition treatment, an intra-specific competition treatment, and then an inter-specific competition. We had 120 plates per treatment, and I monitored individuals from settlement at day 0 up until roughly 140 days post-settlement. So these individuals were monitored in the field. We looked at things like survival, growth, and then reproduction. So that was when they started producing these ovicells. And based on the previous study, this amount of time was found to provide a strong indication of lifetime reproductive output. And then we split our analysis across 3 different fitness measures. So firstly, we had viability selection, which was survival to reproduction, fertility selection, which was the ability to reproduce, and lastly, fecundity selection. So this was the cumulative reproductive output, or the cumulative number of ovicells over the first 20 weeks of the life history. And so for the purposes of today's talk, I'll just talk about the results from the survival and fecundity selection. Okay, so what we found is that there was the overall highest reproductive output in the absence of competition, and the lowest reproductive output at interspecific competition. So we sort of think of these 3 treatments in terms of a stress gradient. So what we found was that the opportunity for selection, so the variance in relative fitness, was 1.5 times higher under interspecific competition relative to no competition. Okay, so I'm just going to walk you through the selection surfaces for the results. So for viability selection, or survival to reproduction, selection on metabolic rates depended on the life history stage. So whether it was 2 or 24 hours post-settlement. And so different combinations of metabolic rates produced the overall highest survival. And so under no competition, we found positive directional selection on metabolic rate late. Under intraspecific competition, I found concave selection, so for intermediate metabolic rate early. But then also across all environments, we found negative correlational selection for metabolic rate early and late. And so this meant that selection favored individuals with either a high metabolic rate early and low metabolic rate late, or vice versa. Okay, so for fecundity selection, which was the cumulative number of ovicells produced, we found that under no competition, there was concave selection for metabolic rate early. Intraspecific competition produced positive directional selection, but only for metabolic rate early. And then for interspecific competition treatments, we found positive directional selection for both metabolic rate early and metabolic rate late. Okay, so just to summarize that, we found that there was greater opportunity for selection under the interspecific competition treatment. Overall, the direction and strength of selection, or the strength of selection, was highest for faster metabolic rates under competition. And then the intensity of selection was greatest in the absence of competition, but we only found this for viability selection. We didn't see any differences in the intensity of fertility or fecundity selection. Okay, so then we wanted to look at how different traits representing the pace of life co-varied with metabolic rates. And so what we found is that higher metabolic rates early and late were associated with faster growth. Lower metabolic rates were associated with a longer lifespan. And then finally, earlier onset of reproduction was associated with a higher metabolic rate late. So just to conclude, competition changed the strength and form of selection on metabolic rates in the field. Competition decreased mean individual fitness, but it increased the opportunity for selection and reduced the intensity of selection. So ultimately, what maintains variation in metabolic rates? Well, we think that variation in selection regimes across small spatial scales are likely to account for this massive variation in metabolic rates within species. So for example, in our case, competition served higher metabolic rates. So higher metabolic rates grew faster and reproduced sooner. So there was selection for higher metabolic rates in the presence of competition, but lower metabolic rates were favored when competition was absent. And I'd just like to thank you for listening, and I hope you enjoy the paper. Thanks.